This recording will focus on the first part of gene function, which is transcription. In order to understand gene function, we have to understand what is the purpose of the genome. Remember that most, if not all, chemical reactions inside a cell happen thanks to enzyme catalysts. Remember from chapter 6, each step in a reaction had its own enzyme catalyzing that step. Also remember that enzymes are made of proteins, and proteins are made of sequences of amino acids. So the sequence of amino acids found in each enzyme is unique to that particular enzyme. In order for cells to be able to synthesize their enzymes, they have to be able to know the sequence of amino acids particular to each enzyme. Therefore, that sequence of amino acids have to be written somewhere as a sort of recipe in the cell. That is the genome of the cell. The genome of the cell is the place where the cell has written the recipe, if you will, of the sequence of amino acids needed to make every single enzyme the cell uses. This recipe is written in genome language, which is DNA language, which is nucleotide language. So the genome of the eukaryotic cell inside the nucleus and the genome of the prokaryotic cell in the nucleoid is the DNA molecule that contains the recipe that will tell the cell the sequence of amino acids needed to make any of the enzymes the cell may need. However, there is a problem. The recipe in the nucleoid, or the DNA molecule in the nucleoid, and the DNA molecule in the nucleus of eukaryotic cells are huge molecules that are coiled and supercoiled and packaged inside the nucleus in the case of eukaryotic cells, or in the nucleoid in the case of prokaryotic cells. This is a huge genome. The cell has to be able to somehow find the recipe for the enzyme it needs, and then it has to be able to make that recipe accessible to the organelles that actually synthesize proteins, the ribosomes. But remember, ribosomes are found in the cytoplasm of the eukaryotic cell and the cytoplasm of the prokaryotic cell. You can see what the problem is, especially in eukaryotic cells. The recipe book, the recipes are inside the nucleus. The kitchen, the organelle that is going to cook or make the proteins, are in the cytoplasm. Somehow the cell has to be able to make accessible that recipe to the ribosomes. Transcription is the process that makes an RNA copy of a gene. Cells need to make a copy or transcription of the recipe or gene in the DNA genome and take that short copy to the ribosomes for synthesis. The copy of the gene is synthesized using RNA. In order to synthesize the polypeptide chains that will make enzymes, ribosomes will need to translate the copy of the recipe. That messenger RNA transcript, which was made in the nucleus or the nucleoid of the cell, is written using nucleotides. Ribosomes will read the nucleotides and translate them into sequences of amino acids that will make the polypeptide chains that will eventually make the enzymes and proteins the cells need. So this is the central dogma of genetics. DNA is transcribed to RNA, RNA is translated to form polypeptides or sequences of amino acids. Transcription will make three types of products from DNA. Ribosomal RNA, which is used to make ribosomes, transfer RNA, which will be used during translation, and messenger RNA, which is the transcript of the copy of the gene that will be translated into a polypeptide chain. So remember that also written in the genome of the cell are recipes or genes to make RNA molecules, transfer RNA and ribosomal RNA. These recipes or products of transcription don't need to be translated. So when you think of the genome, don't forget that genome contains genes for enzymes and proteins, but it also contains genes or recipes to make the tRNAs and the rRNAs the cell needs. The copies of these recipes will not be translated because the recipe is already written in the nucleotide language needed for the final tRNA molecule. Same thing with the ribosomal RNA. The transcript already makes the ribosomal RNA needed. Transcription occurs in the nucleus of prokaryotes, in the nucleus, mitochondria, and chloroplasts of eukaryotes. Essentially, transcription will occur any place where DNA is located. The products of transcriptions will be messenger RNA, which will be translated, ribosomal RNA that will make ribosomes, and transfer RNA, which will be involved in translation. Transcription has three steps: initiation, elongation, and termination. The enzyme involved in transcription is called RNA polymerase. RNA polymerase can only synthesize RNA in the 5' to 3' direction. RNA polymerase does not need a primer. RNA polymerase does not have proofreading ability. The first step in transcription is initiation. Initiation occurs when RNA polymerase binds to a non-coding region of the gene called the promoter region. The promoter region is ahead of the recipe, if you will. A small molecule called a sigma factor will attach to the RNA polymerase. The sigma factor allows the RNA polymerase to recognize the promoter and unwind or unzip the DNA. Remember, DNA is a double-stranded molecule. In order for it to be transcribed, the two strands have to be separated. Now you have the RNA polymerase with the sigma factor binding to the promoter region of the gene. The RNA polymerase with its sigma factors unwind and separate the two DNA molecules. Elongation begins when the sigma factor is released and transcription begins. Notice that transcription will give a single-stranded RNA molecule as the transcript or the copy of the gene. Because RNA polymerase can only make RNA in the 5' to 3' direction, the 3' to 5' DNA strand is the one being used to transcribe the messenger RNA. The 5' to 3' strand below is not being used in transcription. This is an important concept. Notice that the messenger RNA starts with a 5' nucleotide. The last nucleotide will be a 3' nucleotide. Elongation of transcription continues. Typically during transcription, more than one RNA polymerase will transcribe the same gene at the same time. providing multiple copies of the messenger RNA, which can be translated and create multiple copies of the enzyme. Termination of transcription can occur in one of two forms. Self-termination occurs when a hairpin loop is formed at the end of the gene. The hairpin loop forces the RNA away from the DNA strand. Another possible termination strategy is a protein-promoted termination, in which a protein will be present at the end of the transcript, and it will bring the RNA, messenger RNA, away from the DNA strand. Prokaryotic transcription oftentimes transcribes more than one gene at a time. Remember, one gene codes were for one polypeptide. A promoter region ahead of several genes means that when the transcription takes place, the transcript, the messenger RNA produced, will have the copy of gene 1, gene 2, and gene 3. When this messenger RNA is translated, polypeptide 1, polypeptide 2, and polypeptide 3 will be translated. These three polypeptides could combine to make one enzyme. So typically the promoter region is ahead of several genes that have something to do with each other and need to be transcribed at the same time. The messenger RNA transcribed in prokaryotes will not be modified after transcription. The transcription product will be translated. Eukaryotic transcription is different. The transcription product will be modified. Portions of the transcript, of the RNA transcript called introns, will be cut out. What remains will be portions called exons. So introns are portions of RNA that will be cut out of the pre-messenger RNA transcript. The exons will remain. If this is a DNA gene of a eukaryotic organism, the transcription product of that gene contains trash sequences or introns that have to be cut out before the actual coding regions exon 1, exon 2, exon 3, and exon 4 can be ligated together, forming the actual messenger RNA that will leave the nucleus of the cell to be translated. This process is called post-transcriptional modification, and it actually represents an advantage to the cell. If you think about introns and exons as ingredients in a recipe, and you could leave out some ingredients to create a new dish, you can see how cutting out certain portions of a transcript will lead to different enzymes. So within one gene, one recipe, within one gene, the cell could have transcripts for several different enzymes by just cutting out different introns and leaving out different exons. At this point, you should be able to compare and contrast eukaryotic transcription and prokaryotic transcription based on location and mechanisms. Would prokaryotic RNA polymerase be a good target for antibiotics? Or are RNA polymerases, or eukaryotes and prokaryotes, too similar to be targets? During post-transcriptional modification, which are removed, introns or exons? Is there an advantage to post-transcriptional modification? Given the following DNA sequence, choose the appropriate DNA strand to transcribe and give the messenger RNA resulting. Note that transcription proceeds in 5' to 3' direction, so only one of the DNA strands will be transcribed. Also remember that RNA does not use thymine. Instead, it uses uracil.